Alright guys, let's take a look at Lecture 3, Cell Communication. So now we're going to start bringing together all of the parts of the cell that we've learned about, and we're going to talk about how cells use those parts to talk to each other. So you are a collection of about 10 trillion or so cells, and what makes you you is the communication between these cells. So the basis of being multicellular is this communication. And you can either communicate through physical contact, or you can also communicate through chemical signaling. Oops. And I just skipped to my chemical signaling, didn't I? Going back. There we go. So you can also communicate through chemical signaling. So the thing about cells is that cells really depend on each other in order to survive properly. So cells are, here it says social butterflies, right? But but cells really depend on each other to the point where if you isolate a cell from its surroundings and from its buddies, it'll even die. So cells, they stick to each other, they talk to each other, they support each other. And in fact, we have a special name for cells that do this. They're called tissues. A tissue is just a group of cells, all that pull together to do a particular task, and they all do this by communicating with each other. So a couple examples of tissues. Epithelial tissue covers and lines parts of our body. Your skin is epithelial tissue. Connective tissue connects parts of your bodies together. That includes, tendons and bones and things like that. Muscle tissue and nervous tissue. So each of these tissue types will have its own distinct ways of communicating. And we just said there's two main methods of communication: direct physical contact and also chemical signaling. So let's take a look first at direct physical contact. Right? So almost all cells have an outer layer in addition to their cell membrane. So if we think, let me draw a cell here. Here's a cell, here's the nucleus of a cell. We're going to be talking pretty much about eukaryotic cells. In addition to the cell membrane itself, we can also have an additional series of fibers and supportive structures that support and enmesh the cell. And in animals, these extra supportive structures are collectively known as the extracellular matrix. A lot of times you'll see it shortened to ECM. In animals, or rather in plants have a cell wall. And if we recall, that cell wall is made of a polysaccharide called cellulose. In animals, that extracellular matrix is made of a variety of things, and we'll get into that in the next couple slides. So down here is an example of the cell wall. So here's a cell wall of one plant cell. Here's the cell wall of another plant cell right next to it. And there's a little hole in between right there where cells can send signaling molecules, where they can send chemicals through to each other to talk to each other. So let's take a closer look at that ECM, the extracellular matrix in animals. So the ECM provides structural support for animal cells. Your body is actually made, besides water, is actually made mostly of extracellular matrix. So the majority of your weight, again, besides water, is actually ECM. It's not cells. Especially if you look at something like bone, right? Your bones are mostly extracellular matrix. It's those fibers outside of cells. Now, what's the extracellular matrix actually made of? It's made of a combination of these things called glycoproteins or proteoglycans. And if you maybe remember glycoproteins and proteoglycans are made as a result of the secretory pathway. Remember that secretory pathway is where we go from rough ER to Golgi. Alright. So these glycoproteins or proteoglycans make up the majority of your extracellular matrix, and they're spit out by your endomembrane system through the secretory pathway. And these glycoproteins or proteoglycans, there can be many different types of them. The major one that we have in animals is collagen. We have a lot of collagen in our skin, in our bones. We also have another more minor protein called elastin. And we can actually see in the image down here where we see the components of the extracellular matrix. Each little individual dot is a cell nucleus. These dark pink fibers right here, that's actually collagen fibers. And then these more spiny pieces right there, that's actually elastic fibers. So elastic is the name of the fiber, elastin is the protein that makes up the fiber. Now with elastin in particular, you stop making elastin after puberty. So once you hit the end of puberty, you stop producing elastin. And over time, that elastin in your skin and in your bones will degrade, resulting in less stretchy skin and wrinkles and less strong bones. So this extracellular matrix provides a lot of support for these cells that allows them to attach to each other, alright, so they can stick together. It's also what provides cells an environment where they can talk to each other through these things called cell junctions. The cell junctions are made of proteins. And the extracellular matrix is what links the cells inside to its outsides. So let me draw a cell membrane for you really quick, just part of a cell membrane. Inside the cell, remember, we have our cytoskeleton. Outside the cell, we have the extracellular matrix, and the cytoskeleton inside the cell is linked to the extracellular matrix outside the cell via these specialized proteins that link the two. So the inside of the cell is linked to the outside. Here's what some of these collagen fibers look like. So right here, this is a partial collagen fiber. It's a long fibrous protein. And what we do to that collagen fiber and to the collagen protein when the collagen protein's in the rough ER is, remember what we do in the rough ER? We glycosylate. When we glycosylate, we add carbs. We add a long carbohydrate chain, and when we add carbs to a protein, we are producing at this point a proteoglycan, also known as a glycoprotein. So this is what a glycoprotein actually looks like. We have the protein part right there, so that would be the collagen part. And then we have these long strings of polysaccharides hanging off. Remember, polysaccharides and carbs in general are sticky, they interact well with water, so proteoglycans kind of form a gel. And in fact, Jell-O is made of proteoglycan. It's actually made of collagen. Alright, so going back a little bit more to the, if we recall, let me go back a slide, to this part right here where we're talking about linking the inside of the cell to the extracellular matrix. Do I have a picture here? Yeah, I kind of do. Okay. We said that one of the jobs of the extracellular matrix was to hold the cell in place, and the cytoskeleton links, or the membrane proteins link the cytoskeleton with the extracellular matrix, and that's really what keeps cells in place. And so let me again redraw this over here. Here's our cell membrane. Just a partial view of it. Here's our cytoskeleton inside the cell, acting as support. Here's our extracellular matrix outside the cell. Extracellular matrix. And then we have these proteins that link the cytoskeleton with the fibers of the extracellular matrix. Alright, And remember, one of the components of our cytoskeleton is actin. Most often we actually link actin to the extracellular matrix. So we link actin to the ECM using these anchoring proteins, and these anchoring proteins can kind of come in a couple different flavors. Alright, one of them is, and there's several different parts to them. One name for them is an integrin protein. Another name is a fibronectin protein. So these integrins and fibronectins are what actually link to the extracellular matrix fibers, to those collagen fibers. Now, if you break one of these anchorage proteins, if you mutate the gene for it, and I can actually just erase it, can't I? No, I can't. Okay, never mind. Oh, I did. There we go. If we break one of the genes for these proteins, then the cell can no longer anchor itself, and it is free to move. And so if this cell moves, and if it's a cancer cell, it can actually metastasize. Metastasis is where cells move throughout the body. Now, what's also interesting is if you can't produce collagen and you can't produce your extracellular matrix, it can actually lead to diseases of the connective tissue of your bone and of your skin. One such disease is called scurvy. So in order to produce collagen, you need vitamin C, right? And no vitamin C equals no collagen equals scurvy. And some of the symptoms of scurvy include weakened bones. They include your teeth starting to fall out because the collagen fibers that hold them in place no longer are there. And they include sores on the skin, and redness of the skin as your blood vessels start to burst. The collagen fibers holding your blood vessels together start to degrade. So go eat some limes. Alright, get some vitamin C. This is what all this big mess of fibronectin and integrin looks like. So again, we're looking at fibronectin and integrins right here. You don't really need to know this stuff, but just for shits and giggles. Here's our actin filaments on the inside. That's the cytoskeleton of the cell. Here's our fibronectin right there. I'm sorry, that's an integrin molecule right there. Here's our collagen fiber right there. And if we wanted to get excruciatingly close to our fibronectin, here's what it actually looks like right here. It's a disaster. There's multiple subunits here, there's multiple parts to it. So you can imagine pretty easily how something could go wrong here, and that would allow the cell to detach. Okay, so let's talk about some of the connections between cells. So we talked about how cells stick to their extracellular matrix. Now let's talk about how cells actually come into contact with each other and make connections. There are three major connections: tight junctions, desmosomes, and gap junctions. So tight junctions are between tightly packed cells, and these tight junctions, they stitch two cells together to form pretty much a watertight seal. You find these tight junctions a lot in your skin, right? You're waterproof. You find them in the cells that line your intestines. Again, you don't want your intestines spilling out into your body cavities. You find them a lot in your bladder. So what these tight junctions look like is actually on the next slide that we just saw. There's these sort of lacings that link together two cells. So here what we're actually looking at is an intestinal cell. Now some cells will not have tight junctions, but most do in some amount, and the ones that we listed, skin, intestines, and bladder, really have a lot of them. And tight junctions are made of proteins. Again, each little green ball that we see here is actually an individual protein subunit. Excuse me, got the hiccups. And those proteins line up with each other and stick to each other. They bind together. Desmosomes are another one of our cell junctions. And desmosomes, I kind of think of them as like these really, really strong rivets. So they rivet cells together. They really heavily link cells together. They are not waterproof though. So when we look at desmosomes, this is a desmosome right here. Notice how the desmosome is linking one cell's cytoskeleton to another cell's cytoskeleton. There's a lot of strength in here. So desmosomes connect the cytoskeletons of cells together. And desmosomes are made mostly of a protein called cadherins. Cadherins. These are the proteins within desmosomes that actually cause the two sets of desmosomes to stick together. So the cadherins are actually these little green ones in here. Finally, we have gap junctions. Gap junctions are kind of like little holes in the cell membrane. And let me show you gap junctions. Here's gap junctions right here. They're tiny little pores. They allow molecules to very, very easily go from one cell to another cell. So they physically allow nutrients, ions, signaling molecules to go from one cell to the other. Remember most of the time, especially with ions cannot get through the cell membrane. So these gap junctions allow very fast, easy flow of ions from one cell to another cell without having to cross through that pesky cell membrane. This is actually incredibly important in your heart. Your heart cells work together by using an electrical impulse, and they generate that electrical impulse using ions. And if your heart cells don't communicate very quickly with each other through these gap junctions, they can't beat in unison. So you don't generate a coherent heartbeat. And defects in gap junctions have actually been linked to heart arrhythmias, where your heart doesn't properly beat. So if you want to know more about gap junctions and their role in heart defects and other things like that, this is actually a really, really cool article. It's from Scientific American, so it's not terribly hard to read, and it links gap junctions with protein trafficking and, heartbeat and all that. It's pretty cool. Alright, I'm going to hit pause here really quick. I will come back and see you guys in just a little bit. Alright, we're back. Make sure my microphone's working. Yep, we're good to go. So, we finished talking about physical signaling, where cells make physical contact with each other. Now we are getting into chemical cell signaling. So let me maximize this here. And with chemical cell signaling, we need two components. Right? We need two components. The first component is a receptor, and this receptor is almost always made of protein. And then we need the signaling molecule. Okay? So different cells will have different sets of receptors. Right? A neuron will have a different set of receptors than a liver cell, which will have a different set of receptors than a skin cell. And only cells with a particular receptor can receive a particular signal. Okay? So as an example, morphine. Let's use morphine as a particular signaling molecule. Morphine causes neurons to behave in a certain way because neurons have a morphine receptor. That morphine receptor, by the way, also has a name. It's called the mu-opioid receptor. And we thought for a long time only neurons were the ones who had this mu-opioid receptor. One of the really well-known side effects of morphine and a lot of other opiates is actually really severe constipation. As it turns out, cells within your intestines, within your gut, also have the mu-opioid receptor. So morphine was triggering both of these receptors. And just because cells have the same receptor, though, doesn't mean they'll react the same way. Right? So your intestinal cells will react to morphine very, very differently to your brain cells. However, in contrast, your skin cells don't have mu-opioid receptors. They're not going to react at all. When we look at chemical signaling, there's actually three major types of chemical signaling: endocrine signaling, paracrine signaling, and autocrine signaling. Endocrine signaling, we can kind of think of it as long-distance signaling. Another name for it you may hear a lot is hormonal signaling. This is where a cell in one particular location, such as a cell in the thyroid gland or a cell in the pituitary gland, right? These cells create and secrete a particular protein hormone or even a steroid hormone, and they travel through the bloodstream across the body to other totally separate organs. With paracrine signaling, on the other hand, this is a more short distance. This tends to happen between nearby cells, and this is what happens with your neurons actually, in the process known as neurotransmission. So your neurons communicate using paracrine signaling, and paracrine signaling is usually across very, very short distances. So we're talking, micrometers in length sometimes. Autocrine signaling, remember auto means self. This is where a cell tells itself to do something, and a lot of times where this happens is actually in growth. So where this happens is actually in growth. So a cell can actually tell itself in response to maybe more nutrients to actually grow. So what kinds of things do cells signal then? Well, one of the major things they signal is actually growth, as we just discussed, especially after a wound, after cells are damaged, or during embryonic development. You're doing a whole lot of growing during embryonic development. Cells also signal blood vessels to dilate, just another example. So they'll tell blood vessels to open up and your blood pressure to drop. They'll trigger cells to absorb water or to expel water. They will trigger cells to change their metabolism, so to increase their metabolism and to increase the chemical reactions they do or to decrease them. Cells will also signal a change in electrical charge across the membrane. This is actually also how neurons work, is by changing their electrical charges. Same with your heart and your muscles, actually. Cells will also signal more mundane things like color, sound, smell, or pressure, and pain. So remember we said we have to have a signal and we have to have a receptor. Well, the signals can be a particular color light hitting a receptor. It could be a particular sound. Remember, sound is vibration. And it's the vibration that actually triggers cells. It could be a particular chemical triggering smell, etc. So these are all things that, all ways cells that change their behavior in response to certain signals. And then over here, here's an example of a receptor. This right here is the phospholipid bilayer of the membrane. So this particular protein, if we remember our two major protein structures, this is actually a transmembrane protein, meaning it goes all the way across the membrane from one side to the other. And this particular transmembrane receptor is actually called rhodopsin. We'll learn what these GPCRs are in just a few moments. This is a receptor found in the rods in your eye, and they allow us to perceive light. So this is the receptor part, photons would be the signal part. So let's look at the chemical signals first. Okay, so we're going to talk about chemical signals, the signaling part, and then we'll talk about the receptors. There's two major ways we can classify chemical signals. We can classify them as polar molecules, so polar molecules, or non-polar molecules. We often also call polar molecules hydrophilic signals and non-polar molecules hydrophobic. These two terms refer to just their chemical properties. It doesn't refer to how far these signals will travel. We use paracrine, endocrine, and autocrine to refer to how far they're going to travel. So polar chemical signals include any hormone that's a non-steroid. So most of these hormones will actually be derived from proteins. They'll either be fully folded proteins themselves, they'll be short proteins, usually of 10 or so amino acids. Those are called peptides. They can even be single amino acids. Every once in a while you also have other large polar molecules that are considered non-steroidal, but the vast majority of the time you're going to have protein hormones. So protein hormones, hydrophilic hormones, polar chemicals, some signals are polar hormones. All of these, for our purposes, are fairly interchangeable. These guys, because they're so big and because they're so polar, they cannot get across the cell membrane. They kind of have to use a backdoor method, and we'll talk about that. The receptors for them are actually on the surface of the membrane. So let me draw a little tiny membrane here. If you're a polar chemical signal, you can't get across the cell membrane, right? And so you have to have a receptor that goes all the way through the cell membrane from the outside to the inside. It has to be transmembrane. So for our hydrophilic chemical signals, our protein hormones, all those, we're going to have transmembrane receptors. Now for our nonpolar chemical signals, and most of the time these include steroid hormones. If we remember, we actually have two classes of hormones, what are called steroid hormones and peptide hormones. These are our two major classes of hormones. Chemical signals are a broader class that include hormones. So chemical signals include hormones, in which we have our steroid and peptide, and other things such as photons and I should say simple carbon skeletons. So these are just molecules that are a couple dozen carbons. My slide's a mess here, isn't it? Okay. So most of the time with nonpolar chemical signals though, these are mostly going to be steroid hormones. They are by far and away the most common. And these steroid hormones, let me draw a little S here, and other nonpolar chemical signals, they can go straight through the cell membrane. Remember, lipids can go straight through the cell membrane. And because of that, their receptors are actually located inside the cell. So right here, this is an example of a nonpolar chemical signal. In particular, it's a steroid hormone. Okay, this is estrogen. Right here, behind all my chicken scratch, this is called TNF-alpha. It stands for tumor necrosis factor alpha. This is a protein hormone, so it's a polar chemical signal or hydrophilic hormone, hydrophilic chemical signal. Again, all the same thing for us. This actually will trigger your immune system to jump into action. So it'll tell your immune system cells to become more active. Oh, forgot about this part. These chemical signals generically are known as a ligand. Okay, so when a chemical signal interacts with any sort of receptor, any chemical signal, any receptor, that's called that chemical signal is called a ligand. All right, so ligands trigger, as we just said, several different types of responses. What we saw in the previous slide over here, this is just some examples. We can classify our types of responses into three major types. And the first major type is immediately changing how the enzymes in a cell work and how the metabolism of a cell works. All right. So, as an example, we can use adrenaline. Adrenaline is a protein hormone or a peptide hormone. Okay, so it is nonpolar, or it is polar, which means it'll dock with a transmembrane receptor. Adrenaline will dock with transmembrane receptors on cells of the liver, and your liver cells store a lot of glycogen. Now, adrenaline triggers the fight or flight response too. And when adrenaline docks with these liver cells, it'll tell these liver cells, oh, start hydrolyzing glycogen so we can use that glycogen to make ATP, to make energy, because we're getting ready to fight, to punch someone in the face, or to run away. And Besides liver cells, heart cells also contain receptors for adrenaline too, and your heart cells will respond a little bit differently than liver cells. So we can trigger an immediate change in how the enzymes in a cell work and the cell's metabolism. We can also trigger an immediate change in what's called an electrochemical gradient of a cell. And if we don't remember this from Bio 160, here's a cell membrane. Drawn a lot of those lately, right? An electrochemical gradient is where there's a concentration of ions on one side of the cell. Remember, ions can't easily cross the cell membrane, so they're kind of stuck wherever we put them. So this is right here, there's an electrochemical gradient on one side of the cell. And we can actually trigger cells to build that gradient, to concentrate ions on one side of the cell. And when we do that, that actually generates an electrical charge. Neurons do this all the time. We can also trigger a change in gene expression, which genes we use and when. And this tends to be more long-term. It takes a little bit longer. Sometimes, when I say long-term, 20 minutes to a couple days. And this is usually because we are activating or inactivating these things called transcription factors. And if you remember, maybe, transcription factors help with a process known as transcription. And in transcription, we are converting DNA to RNA. Okay. So we can activate or inactivate certain genes by regulating transcription using these special transcription factors. A really cool example of this is actually with estrogen. So estrogen is a steroid hormone. It binds to intracellular receptors inside, let's use as an example, mammary cells. So intracellular receptors. In mammary cells. Mammary. Okay? And estrogen will trigger these cells via their intracellular receptors to actually express different genes, and these cells will build fat as a result. So they'll start making proteins, they'll start reading genes and making proteins involved in building fat. And so these mammary cells will start packing themselves full of fat, and that's why estrogen triggers the development of breast tissue. Here's just some other examples. Okay, so we talked about estrogen, how it was a steroid hormone. It travels inside the cell and it stimulates development of certain female characteristics. Another one is insulin. This is a protein, so this is going to be a large chemical, a large polar signal, protein hormone. It has to dock with transmembrane receptors, and insulin actually triggers us to start digesting glucose. So you do not have to know any of these for this class, but if you end up going into medicine, you will have to know thyroxine, you'll have to know prostaglandins, insulin, follicle-stimulating hormone, and certainly estrogen. All these other ones, ethylene, cysteamine, auxinin, bressin, and steroids, those are actually for plants. Okay, so we talked about how the three different ways that cell signaling can regulate behavior. We talked about some specific examples of changing cell behavior. Let's get into some examples of some of these chemical signals, specifically nonpolar chemical signals, and let's really focus on humans. That's what we're interested in. So many of our steroid sex hormones are nonpolar, right? They're steroids. Okay, these affect sex differentiation, so they cause cells to develop in different ways, and that's what leads to the development of breast tissue and testes and that sort of stuff. And these are almost always endocrine. They're produced by one organ, again, usually the ovaries or testicles, and they travel all throughout the body. Vitamin D is another example of a nonpolar chemical signal. Vitamin D will actually affect calcium absorption from your bloodstream and creation of new bone. So vitamin D actually allows us to absorb calcium from our blood. That's why in a lot of vitamin D supplements, or why on a lot of calcium supplements, I should say, you'll see like now with new added vitamin D or something like that. And this is endocrine. You make vitamin D in part in your skin, and that vitamin D travels to your bone cells. So it travels a long distance. Thyroid hormone is another one. Thyroid hormone affects your basal means baseline or basic. So thyroid hormone affects your baseline metabolic rate. Thyroid hormone is produced by your thyroid gland. It's right at the base of your neck, kind of right above your collarbones. And what thyroid hormone does is it causes us to create enzymes related to metabolism. So metabolism includes things like glycolysis and the TCA cycle and the electron transport chain. So we will start making more enzymes involved in all those processes. And if you have a lot of thyroid hormone being secreted, you're going to start stimulating the breakdown of fat, because you're making all these enzymes that do cell respiration. You're going to break down fat, you're going to break down carbohydrates, dang it, and you're actually going to generate heat. You actually release more thyroid hormone during colder months, so you can keep yourself warmer. In hypothyroidism, so this is where you're producing too little thyroid hormone. This causes impaired memory because you can't break down all of your available energy storage molecules into active energy sources. So your brain doesn't work as well as it should. You're really sensitive to cold because you can't really generate your own heat too well. And of course, you suffer from fatigue just because you can't break down your molecules as effectively as you'd like. So here's what some of these look like. Here's vitamin D right there. That's what it looks like. So that's a signaling molecule. Here's our receptor, the thyroid hormone receptor. Let's look at some polar chemical signals in humans. Adrenaline is a big one. So adrenaline is actually both endocrine and paracrine. You produce adrenaline from your adrenal glands on top of your kidneys. And you release adrenaline in response to fear or anxiety or fright. And we actually have several different receptor subtypes for adrenaline. So as an example, and these adrenaline receptors are all called adrenergic receptors. So adrenergic receptors for adrenaline. Some adrenergic receptors found in smooth muscle surrounding your airways, so your lungs, it causes their cytoskeleton to relax. So your airways dilate, they get bigger. So an adrenaline rush causes your airways to open up so you can breathe a little easier and get oxygen delivered to your cells. Now, in your blood vessels on the surface of your skin, so you have a different type of adrenergic receptor surrounding those in the smooth muscle surrounding those blood vessels. You have smooth muscle surrounding your blood vessels, and this causes the cytoskeleton to contract rather than relax. Different adrenergic receptor, different cell type, different response, even though it is the same signaling molecule. Because the cytoskeleton here contracts, your blood vessels on the surface of your skin constrict. So if you look at someone who's really frightened, they're pale, right? Because their blood vessels constrict. Histamine is a paracrine signal. Histamine is released in response to tissue injury or irritation by those irritated cells. And that actually triggers blood vessel dilation. And it also allows more fluid into your skin cells. So this results in runny nose, right? This results in swelling as that fluid floods in. It results in inflammation, which is redness and tenderness to the touch. And this is all locally. This is what histamine looks like, by the way, down here. This is the histamine receptor. Antihistamines like Sudafed actually prevent your cells from releasing histamine. Bradykinin is one more example. Bradykinin is also released by damaged tissues in response to injury. And it actually will trigger any nearby neurons. It should say stimulates. Stimulates neurotransmission of pain signals by neurons. So bradykinin is a pain molecule. And bradykinin has its own receptor. And this is adrenaline down here. Okay, I'm going to hit pause here. And I'm going to upload this as the first chunk, and I will record the second part as its own chunk.